Hey everyone, Dr. D here, and in this video we are going to be covering Chapter 7 from Talaro's Foundations in Microbiology 12th edition by Chess. So let's go ahead and get started.
Dr. D, 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 Dr. D. Explain stuff.
All right, welcome back to Chapter 7 in our textbook. Let's go ahead and get started here. It's all about microbial nutrition, the process by which chemical compounds or nutrients are acquired from the environment of the microorganism in order to sustain life. So just like you and I need to eat in order to sustain our lives and to grow and for nutrition, so do microbes. And that's what this whole chapter is about. Where do these microbes get their essential nutrients from? When we're talking about bioelements, we're referring to the basic elements required for life. Think carbon, hydrogen, oxygen, phosphorus, potassium, nitrogen, sulfur, calcium, iron, sodium, chlorine, magnesium. These are called bioelements because they're found in living organisms. And oftentimes we refer to these nutrients that organisms need to acquire as nutrients. These are essential nutrients or substances that can be elements or compounds that an organism must get from a source outside the cell. Some of them they require in large amounts. These are known as macronutrients. And some are required in smaller amounts or even in trace amounts. These are known as the micronutrients or trace elements. Do you remember the term organic? An organic molecule? I need you to remember this. An organic molecule is one that contains carbon and at least one hydrogen. So make a note of that. When someone refers to an organic molecule, it's any molecule that contains carbon as well as one hydrogen. So let me quiz you real quick. Is CO2 an organic molecule? Carbon dioxide, CO2. Can you beat Wicket? That's right, Wicket as always. That's right. So no, CO2 is not an organic molecule, right? Because there is no hydrogen in there. So even though it has a carbon, it's not an organic molecule. Whereas methane, for instance, is an organic molecule, and it happens to be an organic nutrient. Carbohydrates. Remember, carbohydrates are made up of some multiple of carbon, hydrogen, and oxygen. Lipids, proteins, nucleic acids. All of these are organic molecules because all of these are made up of carbon and possess at least one hydrogen. Inorganic nutrients are usually atoms, elements, or molecules even, that do not possess carbon and hydrogen. These can include metals and their salts. So here's a bunch of examples that you don't need to memorize, but magnesium sulfate, ferric nitrate, sodium phosphate, oxygen gas, carbon dioxide gas, water. These are inorganic nutrients. Notice that they don't have both carbon and hydrogen. However, in order to grow, organisms need a source of organic nutrients as well as inorganic nutrients. Does that make sense? So we're going to talk about what organic nutrients organisms require and which ones are inorganic and where they acquire them and what they do. A little bit about what purpose those nutrients serve as well. So when we're looking at a cell, remember the cell is the basic unit of life, and many of these microorganisms are made up of just one cell. Well, most of the weight of a cell is called water weight, and that's because 70% of the mass of a cell is basically water, right? The weight of a cell is water. However, if you were to dry the cell out completely, that would be called dry weight. And 97% of the dry weight of the cell is made up of these organic compounds, right? These molecules that have carbon and hydrogen. And about 50% of that is proteins. And did you know those biomolecules that make up your cells are made up of basically 6 elements. That's carbon, hydrogen, oxygen, phosphorus, sulfur, and nitrogen. So you may want to remember these because these are important. In fact, I have a little acronym here that reminds me of these important elements. I call it CHOMPS, right? So CHOMPS, CHOMPS is great because it reminds me that biomolecules contain carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. So here you can see the biomolecules are built with CHOMPS, right? So here you can see proteins. Do you know the 4 classes of biomolecules from Bio 1? Proteins, nucleic acids, remember that includes the DNA and the RNA, carbohydrates, and lipids, right? These are known as the 4 classes of biomolecules. And remember, 97% of the dry cell weight is organic molecules, right? And these are the 4 big classes of biomolecules, right? These are the main composition of that 97% dry weight, right? And what do we see here? Look, proteins, remember, proteins are made up of amino acids. And what elements make up an amino acid? See, these are both amino acids, right? There's 20 different naturally occurring amino acids. Well, guess what? Amino acids have carbon, hydrogen, oxygen, nitrogen, sulfur, everything except phosphorus, right? The nucleic acids have carbon, hydrogen, oxygen, nitrogen, phosphorus. You see? The lipids can have carbon, hydrogen, oxygen, and phosphorus, like this phospholipid down here. And look, do you remember what I said carbohydrates are? Some multiple of C, H, and O, carbon, hydrogen, or oxygen. So notice that CHOMPS, right, really do make up your biomolecules, the 4 classes of biomolecules. And so these are obviously macronutrients, nutrients you need in large amounts, right? Because they make up 97% of the dry mass of your cell, right? So again, carbon, so important. It's found in nature. Some organisms capture it as CO2. Some organisms capture it as carbonate. Some organisms capture it as organic compounds, like you and I capture it as an organic compound. When we eat food, don't we eat sugars? Don't we eat fats? Don't we eat proteins, right? Those are called organic compounds, right? And by the way, I'm going to go into a lot of detail about carbon, but I don't need you to know everything about carbon. I don't need you to memorize everything on this slide. It's just I'm going to present it to you so you understand. But, obviously you don't have to memorize all the stuff on these slides. It's too much to memorize. But obviously, if you're an organism, you need a source of carbon. Isn't that neat? So where do you and I get our carbon? We get ours from organic compounds in our food, don't we? But did you know, let me make you think a little bit. What about plants? Do plants eat organisms? Do plants eat food? Do plants get their carbon from organic compounds? That's right, Wicket. Wicket chimes in even when he's not even asked. No, did you know that plants and algae get their carbon from the air, right? In the form of CO2. It's called carbon fixation. They literally take carbon out of the air. And that's why they're so good at combating greenhouse gases, right? That's why they're so good at, helping reduce CO2 in the atmosphere, because they literally take CO2 out of the air and they use that to build things, right? Build organic molecules that they need. Isn't that neat? So plants get their CO2 from the air. You and I get our carbon from eating living things. And obviously, we need carbon in order to build our own organic molecules so that we have something that we can use to build our own proteins and such. And also, we eat so that we can have energy as well, right? We eat sugars and fats for energy. So carbon's so important. Next, how about nitrogen? Nitrogen comes from many different sources. For you and me get our nitrogen from organic nitrogen, right? By eating proteins, by eating nucleic acids, that's how we get our nitrogen. But did you know that microbes can get their nitrogen from the soil, right? Think about nitrate, the chemical, or nitrite, the chemical, or ammonium, the chemical. Some plants can even get nitrogen. Some organisms too can get their nitrogen from the air. It's called nitrogen fixation. Just like these guys, just like plants could take CO2 out of the air, literally take CO2 out of the air and capture that carbon, some organisms can take nitrogen out of the air and capture that nitrogen too. So, obviously, if you're an animal, you're eating your nitrogen, right? But if you're a bacterium that lives in the soil, you can get it in one of these forms, or you could even capture it out of the air. It all depends on which organism you're talking about, right? But these are all sources of nitrogen for the different forms of life on Earth, if that makes sense. And obviously, remember, we need nitrogen in order to build our proteins, in order to build our nucleic acids. We need nitrogen. It's one of the chomps, right? So what about oxygen? Obviously, you and I need oxygen. We get our oxygen as gas, right? O2 gas. Okay, but obviously there's oxygen in the air, there are oxides, there's water. These are all sources of oxygen. In fact, the air you're breathing right now is about 20% oxygen, right? It's mostly nitrogen, but it's about 20% oxygen. Hydrogen, there are many different sources of hydrogen for organisms. You and I, obviously, we get our hydrogen from organic compounds. So think about when we eat sugars and fats, those hydrogens are found on the sugars and the fats. But guess what? Some organisms can capture hydrogen gas. They can capture it from water. In fact, plants obtain their hydrogen from water. They take water and then they take the little hydrogens away from water in order to do photosynthesis. Isn't that neat? Some organisms can take hydrogens from hydrogen sulfide. Some can take hydrogens from methane. It all depends on the organism. Isn't that neat? But we all get what I'm trying to get across to you is that we all the life on Earth, they require chomps, right? But they get them in different places from different sources. And that's really showing you the diversity of life. Not everything lives like you and me, like animals, right? Not everyone just consumes other organisms to get their biomolecules. Some organisms can just take those inorganic molecules out of the soil or from the air. And by the way, in case you're wondering, hydrogen is important in cellular respiration. It's important for photosynthesis. It's important to maintain pH balance. So, you do need a source of hydrogens, as well as the fact that those hydrogen molecules are part of the biomolecules. Remember the 4 classes of biomolecules as well. Phosphorus. Remember, phosphorus is so important. It's found on DNA and RNA and some lipids. And do you remember ATP? You ever heard of ATP, the power, the energy currency of a cell? Well, TP means 3 phosphate groups. So you need a source of phosphorus in order to have phosphates, right? Because phosphates are essentially phosphorus with 4 oxygens, right? And so usually organisms get them from nature, from the rocks and mineral deposits. Organisms get it from their environment or from the soil. You and I obviously get our phosphate from our diet, from what we consume. What about sulfur? Sulfur is important. It's found in a couple of the amino acids. Methionine and cytosine include. Those are amino acids that have sulfur in them. Obviously, you don't need to memorize that, but so there are, reasons that sulfur is required in your diet. And so you and I obviously get our sulfur from eating other organisms, but you can find it in nature as just sulfur, sulfate, sulfhydryl. You can find it in mineral deposits. You can find it in sediments. You can find it in soil. And so, like I said, all organisms need sulfur, and we just get it in different ways. Potassium's so important. We get it from our diets, but organisms can get it again from mineral deposits, ocean water, soil, and it's so important in membrane synthesis. And it's so important in membrane transport as well as protein synthesis. It's such an important element. Sodium's another one of these important elements for membrane transport, membrane actions, and it's found, again, in you and me. In animals, it's found in our diet. In fact, we get everything from our diet, really, from consuming, but different organisms can get it from the soil or from the minerals in which they live. Same thing with calcium and magnesium. Again, organisms usually find these elements from their environment as well, and these are very important elements in the functioning of the cell. Same thing with chlorine. Usually these organisms find it in their environments. Again, it's so important in membrane transport. It's important to regulate osmotic pressure. It's an important element for cells. Zinc and iron are important metals. They are usually used as cofactors, which are important enzyme helpers, and they're also found in the organism's surroundings. And then the other micronutrients or trace elements. By the way, do you know what micronutrients or trace elements are called in your diet? Do you ever think about that? They're called minerals, right? Have you ever heard of vitamins and minerals, right? That your diet needs vitamins and minerals. Well, did you know that vitamins are organic nutrients that you need to consume, and minerals are inorganic nutrients that you need to consume, right? These are known as cofactors. Vitamins are organic cofactors. Minerals are inorganic cofactors. What are cofactors? Enzyme helpers. These are substances that help your enzymes function. Next, this is a term that we're familiar with already from a couple of previous chapters. Remember that term heterotroph? It refers to how organisms get their carbon. Heterotrophs, if you remember from the previous chapters, they get their carbon from an organic molecule. So basically like you and me do, right? By eating organic molecules. So they consume proteins, carbohydrates, lipids, nucleic acids, other living organisms. So what you can say are animals and animal-like organisms, the consumers in the ecosystem, are called heterotrophs, okay? Plants, on the other hand, do you remember plants? Where do plants get their carbon from? Do you remember, and can you beat Wicket? That's right, Wicket, as always. CO2, right? Remember I told you that plants get their carbon out of the air, right, as CO2? So that would be called an autotroph. An autotroph is an organism that uses CO2 as their carbon source, right? So it's an inorganic carbon source. And plants, algae, right, photosynthesizers are autotrophs usually, right? Whereas animals are heterotrophs. Does that make sense? So when you think of heterotroph and autotroph, think of where do organisms get their carbon. Heterotrophs get it as by eating organic molecules, right? And autotrophs get it through by getting that CO2, inorganic CO2, out of the air. And do you remember when I said that minerals are inorganic cofactors? Well, the organic cofactors are called vitamins, remember? And in fact, vitamins are a group of a bigger classification known as growth factors. Growth factors are organic molecules that need to be provided to the organism for survival. So this includes vitamins as well as essential amino acids as well. These are organic molecules that are required for growth. Now here's an important table that refers to nutritional types of organisms. Do you remember that we classify organisms based on where they get their carbon? Those are known as autotrophs and heterotrophs. We already covered this. Autotrophs get their carbon as CO2. Heterotrophs get their carbon from reduced, preformed organic molecules from other organisms, basically from organic molecules. But we also classify organisms based on their nutritional type for where they receive their energy from and where they get their electrons from. Energy, let's talk about Phototrophs versus chemotrophs. Take a look at this. Phototrophs are organisms that get their energy from light. So think plants, think about algae. Chemotrophs are organisms that get their energy from either organic or inorganic compounds. So basically some kind of chemical, some kind of molecule, right? They get their energy from that. So let me ask you this. Which one are we? Are we phototrophs or chemotrophs? Can you beat Wicket? Yeah, obviously we can't photosynthesize, right, Wicket? So we are chemotrophs. We get our energy from the molecules we eat, like sugar, like fat, right? I'm ingesting these organic compounds, and that's what's giving me energy. Next, we also classify organisms, nutritionally based on the electrons. Like, where do they get their electrons? And you might be wondering, why do we need electrons? Why do organisms need a nice source of electrons? Well, do you guys remember the electron transport chain? You ever heard of that thing, the electron transport chain? We're going to cover it later this semester. But the electrons are used in the electron transport chain during either photosynthesis uses electron transport chain, the cellular respiration, the mitochondria has an electron transport chain. So anytime an electron transport chain is needed, either for cellular respiration or for photosynthesis, electrons are needed to do the electron transport chain. Does that make sense? So organisms need an electron source. And so lithotrophs are organisms that get most of their electrons from a reduced inorganic molecule, whereas organotrophs get their electrons from organic molecules. And so you and I, for instance, we eat sugar, which is an organic molecule, and fats, which are organic molecules, and we take those electrons from those molecules. Does that make sense? That's where we get our electrons from for the electron transport chain. So we are organotrophs. Plants, on the other hand, did you know that plants need electrons too for photosynthesis? And did you know plants take electrons from water, right? So is water inorganic? Yes. So plants are lithotrophs because plants get their electrons from water, right? And water's inorganic. Whereas you and me are organotrophs because we get our electrons from organic molecules. Does that make sense? Now let's combine all these terms, okay? Did you know that you can combine all of these terms in order to define a nutritional type for an organism? So for instance, plants and other photosynthesizers are called photolithoautotrophs. Photolithoautotrophs. Wow, that's a mouthful, but don't get overwhelmed by this term. Instead, look at this table to break it down, right? So a phototroph is an organism that gets its energy from light. Litho means an organism that gets its electrons from an inorganic source. And auto means an organism that gets its carbon from CO2. So photolithoautotrophs, that's the way they work. That's where they get their energy, their electrons, and their carbon, right? Isn't that neat? So when you see these big long terms, that's what it is. It's basically a amalgamation of all these different terms here. So a plant is a photolithoautotroph, right? A plant is a photolithoautotroph because it gets its energy from sunlight. It gets its electrons from water if it's an oxygenic photosynthesizer. Did you know that plants, algae, and cyanobacteria, these are known as oxygenic photosynthesizers because they make oxygen. The oxygen you're breathing right now is thanks to these little oxygenic photosynthesizers. And did you know the reason they make oxygen is because they take water and then they take water's H's away, right? If you take water's electrons away, you're left with oxygen. That's why plants make oxygen. Isn't that neat? But there are also photosynthesizers that are anoxygenic. You know what that means? It means an organism that does photosynthesis, but it doesn't make oxygen. So in this case, they're probably not using water as their electron source, but they're using some other inorganic molecule as their electron source. So for instance, the purple and green sulfur bacteria, they might use molecules other than water as their electron source. And if you don't use water as your electron source, then you're not going to release oxygen, right? Does that make sense? I hope that makes sense. Again, you don't need to know all this, the level of detail, like that level of detail, but you should know a photolitho autotroph, what that means, and you should know that some organisms, some photosynthesizers produce oxygen, and believe it or not, some photosynthesizers don't produce oxygen. Isn't that neat? Now, this is a strange creature, right? There are some creatures that are chemo, litho, autotrophs. You know what that means? They get their energy from a molecule, some kind of compound, not the sun. They get their electrons from an inorganic molecule, and they get their carbon from the air. Isn't that neat? So for instance, that wouldn't be you and me, would it? That's not humans. But methanogens, this is a type of archaea, right? These archaea are a kind of chemolithoautotroph that produce methane gas under anaerobic conditions. Isn't that neat? So they produce methane gas. In fact, do you know what these guys do? They take CO2 out of the air, right? Because they're autotrophs. They take CO2 out of the air, and then they convert the CO2 to methane, right? Isn't that neat? And they produce methane gas. It's not the best because methane gas is a greenhouse gas, but these guys produce methane. That's what they produce as a byproduct. And they get their electrons not from sugars and fats or an organic source, but some kind of inorganic source, such as hydrogen sulfide or something like that. That's why you can often find these creatures in sulfur springs and sulfur vents, or places that are rich in whatever inorganic electron source they use. Now, this one's near and dear to our hearts, right? These are known as the chemo-organo-heterotrophs. What does that mean? You get your energy from some kind of molecule. You get your electrons from an organic source, and you get your carbon from an organic source as well. So we are chemo-organo-heterotrophs. That's what animals are. Did you know that? Because we get our energy from the chemicals we eat. Those are organic chemicals that give us our electrons. Remember, sugars and fats are where we get our electrons from. And then we are heterotrophs. We also get our carbon from those organic molecules we eat as well. So when you think animals, you should think chemo-organo-heterotrophs. When you think plants, you should think photolithoautotrophs, right? So I hope that makes sense. And do you remember that fungi are saprobes, free-living microorganisms that feed on organic detritus from dead organisms? So the saprobes would be an example of a chemo-organo-heterotrophs. Also, parasites. Parasites are examples of chemo-organo-heterotrophs as well, usually. Here you can see how a saprobe breaks down debris, right? Organic debris. So this would be the fungal cell, for instance, or the saprobe. The saprobe releases enzymes outside of the cell wall. Isn't that neat? So it releases enzymes outside of the cell wall. Those enzymes break down dead organic material outside, producing these food particles. And then those food particles are absorbed and the nutrients are absorbed. Isn't that neat? So this is how saprobes get their energy. All right, concept check. Number one, let's see if we can figure this out together. if an organism is degrading large organic molecules, right? So that means it's an organotroph, to get both carbon and energy, right? Carbon meaning you are a heterotroph, energy meaning you are a chemotroph, right? So which one would you be? Oh yeah, C, right? A chemoorganotroph, right? So the answer is C. I hope you can break it down that way. It'll take some practice, but I think you can nail it. In this next section, we're going to talk about how transport occurs across the cell membrane. Essentially, how do things get into the cell? How do things get out of the cell? Is energy required or not? So we're going to start with this discussion of passive transport. Passive transport is when substances cross the membrane of a cell without any energy requirement, without any energy input. So to fully understand passive transport, we need to understand diffusion, because that's what's driving all passive transport. All passive transport depends on diffusion. And then we're going to talk about a type of passive transport called osmosis, which is essentially the diffusion of water across a membrane. And then we're going to talk about facilitated diffusion, where solutes that require a carrier or transport protein diffuse across a membrane. So hopefully by the end of this section, you'll understand all these terms: diffusion, osmosis, facilitated diffusion. And hopefully you've had this before, you've studied diffusion before, but if not, fret not. I do have a video that I'll link in just a minute. I'll throw up a card above me, which explains how osmosis works in different types of cells. And then conversely, if passive transport is not happening, that means that it requires active transport, right? Active transport is where energy is required in order to move something in or out of the cell. And the big example, there's many forms of active transport, but the one we're going to focus on is called bulk transport, endocytosis, exocytosis, right? We're going to focus on those. So to start, let's talk about diffusion. Diffusion is a spontaneous process. That means that no energy is required for diffusion to occur. It's just going to occur. In fact, diffusion releases energy, if anything. So here it says in your notes that net movement of molecules down their concentration gradient by random thermal movement is known as diffusion. That's a fancy way of saying that substances move from areas of higher concentration towards areas of lower concentration. That's what they mean by moving down their concentration gradient. So look at this. Here's some substance inside of this beaker of water. This substance is high in concentration right here and low in concentration near the edges of the beaker. That means that diffusion will occur. The substance will diffuse from areas of higher concentration at the center of the beaker to areas of lower concentration near the edges of the beaker, right? So that's what moving down the concentration gradient means. A substance always moves from an area of high concentration to an area of low concentration. And that's diffusion. That's what they mean by diffusion. And this occurs until those substances are equally spread out throughout the environment, and that's known as equilibrium. And by the way, this is a process that's spontaneous. It's passive. It releases energy. It does not require energy for diffusion to occur. Now, do you remember, these are two words that you need to write down, okay? Write down these two words, solute and solvent, right? Do you guys remember those two words, solute and solvent? The solute is a substance that gets dissolved inside of a solvent. So if I have a solution of something, let's say I have sugar water, right? Let's say I have sugar water. That's a solution, right? And that means that there's a solute and a solvent. The solute would be the sugar, and the solvent would be the water. Does that make sense? So think of the solvent that dissolves the solute. Now, think of a membrane. If water is what's diffusing across the membrane, that's what they mean by osmosis. That's all osmosis is. It's the diffusion of water across the membrane. If it's the solute that's diffusing across the membrane, that's different, isn't it? Right? If it's the sugar that's diffusing across a membrane, that's not osmosis. That's because that's the solute, right? Does that make sense? That's called dialysis. That's a different concept, right? So osmosis is the diffusion of water across a membrane from high concentration to low. Dialysis is the diffusion of solute across the membrane from high concentration to low. So that's what we need to understand about osmosis. Oh, and before I forget, osmosis always wants to proceed in the opposite direction to dialysis. So let me put it this way. If water wants to go this way across a membrane, that means that the sugar always wants to go the opposite way. The solute always wants to go the opposite way. Whichever way osmosis wants to proceed, dialysis wants to proceed the other way. The solute always wants to go in one direction. The solvent always wants to go in the other direction until everything's balanced out, until equilibrium is reached. So what happens in cells is that living membranes, the membranes of cells, permit free diffusion of water. That means water can enter the cell, water can leave the cell, and this is known as passive transport because it's a type of energy-free process through osmosis, right? Through pore proteins called aquaporins. These little protein pores, basically they're little holes that let water into the cell and out of the cell, right? And so water can flow into the cell, water can flow out of the cell. And when concentrations of solutions differ, one side will experience a net loss of water and the other a net gain of water until equilibrium is reached. So one thing you need to understand is that when you place a cell into a solution, okay? When you place a cell into a solution, water can enter the cell, water can leave the cell, but the solutes stay put, right? Does that make sense? So let's say you place a cell into super sugary water, right? You would think, well, then sugar should get into the cell, right? Sugar so saturated in this environment, sugar should enter the cell. That's not the way it works, right? Water is what moves into or out of the cell, not the solute. So think about it this way. When you place a cell in an environment, it's the water which will either enter the cell or leave, not the solutes, not the sugars, not the salts, not the other substances. Not the ions, right? Does that make sense? I hope that makes sense. So here's an example of osmosis. Remember, osmosis is the passive transport of water. Here you can see this is showing you a bag, okay? And this bag is full of a solution of sugar water. The pink or red dots represent solutes. So let's say sugar, for instance. The red dots are sugar, okay? And the blue dots are water, right? So in this case, take a look. The beaker itself has no sugar in it. So it's 0 solute, 100% solvent, 100% water, right, in the beaker. Look in the bag now. The bag has a high concentration of sugar, right? It has water, but it has a lot of sugar. So again, this is an example of a osmosis problem. Do you know why? Because I want you to take a look at the membrane of this bag. I want you to look at this white line, the membrane of the bag. And if we zoom in on it, what do you notice? You've got tiny little pores. Now let me ask you this. The water seems to cross the little pores, right? but can the sugar molecules cross these pores? Can the sugar get across the pores in the bag? No, right? It's because the sugar is too big to cross the membrane. This would be an example of a semi-permeable membrane. All right, so that's the semi-permeable membrane, right? So that means that the solute can't leave. So yes, students say, well, the sugar should leave the bag, diffuse out of the bag, right? Well, yeah, it wants to, but it can't. Does that make sense? And anytime the solute can't get out or can't get in, can't cross the membrane, that makes it a osmosis problem, because that means only the solvent, the water, is what's going to move across the membrane. And here's the tricky part, right? This is the tricky part, and I'm going to make it less tricky. And I just want you to focus in on this right here. Maybe write this down, okay? When there's passive transport happening, okay, during passive transport, every substance diffuses down its own concentration gradient. I want you to write that down. Every substance diffuses down its own concentration gradient. Do you know what that means? That means, for instance, look, the sugar. Where's the sugar high in concentration? In the bag. And where's the sugar low in concentration? In the beaker, right? So that means that the sugar would like to diffuse from where sugar is high in concentration to where sugar is low in concentration. Does that make sense? Does that make sense? So sugar wants to go from where sugar is high to where sugar is low, right? But can sugar go from where sugar is high to sugar is low? In this particular example, can you beat Wicket? No, that's right, Wicket. Now, remember, the pores are too small. So in that case, remember, the water is what's going to move. This is the part I want you to pay attention to. That means that if water is what's moving, does this make sense? Watch this. If water is what's diffusing, if water is what's moving, water will go from where water is more concentrated, from where water is more pure, to where water is less concentrated, to where water is less pure, right? So if that's true, which way should the water net move, right? Should the water flood into the bag or out of the bag, or neither, right? Can you beat Wicket? That's right, Wicket. As always, water should go from where water is more concentrated in the beaker towards where water is less concentrated in this bag, right? Does that make sense? So let's see if that's what happens. Yes, look, it looks like water is net diffusing into this bag, and that's through what process? Osmosis, because that's what osmosis is. Osmosis is the diffusion of water across a membrane, right? Not diffusion of solute. That would be called dialysis. And so exactly, so water keeps rushing in and trying to balance out the solution, but it'll never balance out. Why? It'll never balance out. Well, because, you're never going to have an equal concentration of sugar inside and outside, because there is no sugar outside, right? Does that make sense? So water will keep rushing in, keep rushing in, keep rushing in until it physically can't rush in anymore. Does that make sense? That's pretty much the way it's going to work. So when we're talking about osmosis happening to cells, those are called tonicity problems, right? Anytime you see the term tonicity, you should think about a cell being in an environment where water either rushes into the cell or rushes out of the cell, okay? And I'm not going to explain exactly how that works, because it all works. It all works exactly like what we just went through, right? If there's a higher solute concentration inside of the cell, then water will rush into the cell. If there's a high solute concentration outside of the cell, water will rush out of the cell. If there's an equal concentration of solute, let's say sugar, for instance, inside and outside of the cell, well, then there will be no net flow of water either into the cell nor out of the cell. And that's exactly what happens when you place cells into different solutions, right? So to explain this, again, like I said, I have a whole little video. It's a short video on exactly how tonicity works, right? So I want you to watch that because, I don't want to make this video longer than it needs to be, especially if you studied tonicity in 1406, in Biology 1. So right here above my head, I want you to click on that card, and that is a short video from me on exactly how these problems work. Like, when does water rush into a cell? When does water rush out of the cell? And why does that all happen, right? How do these tonicity problems work? What happens in an isotonic environment? What happens in a hypotonic environment? What happens in a hypertonic environment, right? So what happens when you don't have a cell wall and you're in an isotonic environment, or if you don't have a cell wall and you're in a hypotonic environment, or if you don't have a cell wall and you're in a hypertonic environment, right? So these are 6 different scenarios that I explain in that video that I had linked above, right? So make sure you watch that before you move on with the rest of this chapter. Okay, I'm assuming you either watched that or you know about tonicity, so let's carry on. If we have cells, so let's talk about cells with cell walls, right? Cells with cell walls. This means that you have some kind of cell wall outside of the cell membrane, like, like fungi do, algae have cell walls, right? We have a cell wall, okay? In this case, in an isotonic environment, water is going to neither net flow into the cell nor net flow out of the cell, right? Because the solute concentration is already equal inside of the cell and outside of the cell. That's what an isotonic solution is. An isotonic environment is an environment which matches the solute concentration of the cell. That means that for every 1 water molecule that enters the cell, 1 water molecule leaves the cell, and so the cell does not net gain water, nor does it net lose water. So you should say that the cell, it does not gain nor lose water. However, if I were to place a cell that has a cell wall in a hypotonic solution, a hypotonic environment, this means that it's a high water environment, a low solute environment. So imagine if I put it, for the best example is if I place a cell in tap water, right? Just pure or distilled water. That means that there is no solute in the environment. A hypotonic environment is kind of like this. You see, remember this example here? This is a hypotonic environment because look, it's only pure water outside, or the water outside of the cell is more concentrated than the water inside of the cell, right? So think of tap water as a hypotonic environment. Again, the solute can't move, and so the water will move from where water is more pure to where water is less pure. And this forms what's known as a high pressure cell because there's a net flow of water into the cell. There's a net flow of water into the cell, but does the cell rupture? No, because the cell wall prevents the cells from bursting. These are known as, by the way, turgid cells. Turgid cells are high pressure. Conversely, if I place a cell that has a cell wall into a hypertonic solution, this means a high solute concentration solution. So imagine if I place an algae cell, into super salty water or super sugary water. That would be a hypertonic environment, a very solute rich environment, either sugar or salt or some other solute. In this case, remember, the solute doesn't move. Water will flow out of the cell because water will be more pure inside of the cell than outside of the cell. This means that water will diffuse out of the cell. Water will go from where water is high in concentration down its concentration gradient to where it's low in concentration. So this means that water diffuses out of the cell and shrinks the cell membrane away from the cell wall. By the way, look at this. The cell wall is too rigid to shrivel. Look at this. The cell wall is like, think of like a brick wall, right? It's not going to shrivel. But what does shrivel is the plasma membrane inside. See this? The plasma membrane peels away from the cell wall. Let me show you something. Here, notice how the plasma membrane is pushed up against the cell wall, right? But well, in a hypertonic environment, so much water leaves the cell that the plasma membrane shrivels and peels away from the cell wall. Isn't that neat? And this is a state called plasmolysis. And microorganisms really do not like to be in plasmolysis. They're getting dehydrated. They're dying at this point. Now, what if you don't have a cell wall and you find yourself in all these environments? Remember, an isotonic, hypotonic, hypertonic environment. Let's talk about that. So let's say you're a little protozoa like Paramecium, and you're in a, you don't have a cell wall. You don't have a cell wall. You obviously you have a cell membrane because all cells have a cell membrane, but you don't have a cell wall, right? In this case, if you find yourself in an isotonic environment, that means an environment with the same solute concentration as the cell. Which way should the water flow? Can you beat Wicket? That's right, Wicket as always. Water should not net flow into the cell. Water should not net flow out of the cell, because think about it. In an isotonic environment, that means you're already at equilibrium. There's no mismatch. There's no higher water concentration on either side of the membrane. Does that make sense? So in this case, for every one water molecule that enters the cell, one water molecule leaves the cell. So, the rates of diffusion of water are equal in both directions. Now, what if I put my Paramecium friend into pure water or something like that, right? That would be called a hypotonic environment. A hypotonic environment, like I said, the best example is like pure water. But it's a hypotonic environment is any time you have a lower solute concentration outside of the cell versus inside of the cell, right? So in this case, water will flow which way? Water will flow from where water is more concentrated outside of the cell into the cell. That means that there will be a net flow of water into the cell. And look what happened. What happened? What happened? So much water rushed into the cell that it lysed, osmolysis, right? That means lysis due to osmosis, osmolysis. So this means that the cell lysed, the cell burst. And the cell burst because there was no rigid cell wall holding it together. Isn't that neat? So if you are a creature that lacks a cell wall, well, then you're going to be more susceptible to osmosis, right? To what's called osmotic shock. This is what they mean by osmotic shock, right? It's like osmosis causing damage to the cell. Isn't that neat? Now, what if I took my Paramecium friend that doesn't have a cell wall and I placed him in a super salty environment or a super sugary environment, right? So that would be an example of a hypertonic environment. And can you beat Wicket? Which way would the water flow? Yeah, out of the cell. That's right. Water would be more pure inside of the cell versus outside. That means water would net flow out of the cell. And this time the whole cell shrivels, right? The whole cell shrivels up. You may have learned this as a crenated cell or a shriveled cell. The cell becomes shriveled and shrunken. Now, you may have heard of facilitated diffusion. This means that a facilitator or a transport protein is required in order for diffusion to occur across the membrane. And by the way, remember when I told you that... Do you remember when I told you that during osmosis, when When water's entering the cell or exiting the cell, didn't I tell you that the water is flowing in and out through proteins called aquaporins, right? The water's not going directly through the cell's membrane. The water's flowing through these little water pores, aquaporins. That by itself is an example of facilitated diffusion because you have a protein facilitator, right? Without this aquaporin, the diffusion wouldn't occur across the membrane. So this is called facilitated diffusion. So let me show you exactly what I'm talking about. Look at this. Let's pretend this is the cell's membrane in brown, okay? If a substance moves from high concentration to low, if it crosses the membrane directly like this, look, if these purple structures were to cross the membrane directly across the membrane, that's just called simple diffusion, which is a type of passive transport. However, remember what I said, that water needs a protein to cross? Well, if you need a protein to diffuse across the membrane, that's called facilitated diffusion. Does that make sense? Facilitated diffusion requires a transporter for the substance to diffuse across the cell by passive transport. And if you have just regular simple diffusion, then you don't require a protein in order to cross the membrane during passive transport. And remember, with active transport, it requires energy in order to cross the membrane. And there are many different types of active transport, but we're going to talk about endocytosis and exocytosis, right? Those are the ones we're going to talk about, starting with what's called endocytosis. Endocytosis means bringing substances in through the membrane. And this is either going to result in bringing in solids, which is called eating or phagocytosis, or bringing in liquids, which is called drinking or penocytosis. Phagocytosis is cellular eating, penocytosis is cellular drinking, and this is the way that substances are brought into the cell in bulk, right? These are both, endocytosis is a type of bulk import into the cell. And you really see this nicely with amoeba, right? Amoeba that have pseudopods. They'll wrap their little membranes around food and bring them in inside of vacuoles. Remember, we kind of touched on that in a previous chapter, forming the phagosome, which fuses with a lysosome, and then you digest that food that you internalize. Remember that from a previous chapter? So let's take a look at them. Let's take a look at phagocytosis. Imagine if this right here in brown, this is an amoeba, right? Like a protozoa. And look here, this is some kind of food, right? These are food particles or something you want to digest. See what happens? The food touches the membrane. The membrane invaginates, which means it kind of folds inwards, and then it wraps around. The membrane wraps around the food. And basically think about this, like extensions of the plasma membrane wrap around the food until they pop, right? They bring in a part of that membrane. Part of the membrane pinches off and comes in with the food inside. Remember, this is called a food vacuole, or sometimes called a phagosome. And then, do you remember, this will fuse with a lysosome, which is full of hydrolytic enzymes, which will chop up the food and prepare it for, digestion, for nutrition, right? So do you see how this brought in a solid? This brought in solid food. That's called phagocytosis. However, what if the amoeba brought in liquid, right? Look, in this case, it's using its membrane to bring in a vesicle full of liquid. So you brought in liquid, or you brought in solution from outside. Well, that's called penocytosis. Penocytosis is bringing in liquids. That's why they call it drinking. Phagocytosis is bringing in solids. That's why it's called eating, right? But both require energy, and both are examples of endocytosis, because your endocytosis means bulk import into the cell, right? So both phagocytosis and penocytosis are examples of endocytosis and both examples of active transport. And by the way, exocytosis would be the opposite. A vesicle full of waste or some kind of other substance will originate from the cell. It will fuse with the plasma membrane and it'll release all of its contents. So imagine this in reverse. That's exocytosis. Exocytosis means bulk export, right? So that's what exocytosis is. And that also requires energy as well. All right, and that leads us to concept check number 3. If a cell is in a concentrated glucose solution, very high glucose solution, and the glucose is moving into the cell through a carrier, right? So it's doing facilitated diffusion. This would be an example of facilitated diffusion. Remember, the glucose is crossing into the cell, but is it going into the cell directly through simple diffusion? No, it's going through a carrier protein. A carrier protein is a protein that allows this solute to cross in. So remember, that means facilitated diffusion. Does that make sense? I hope that makes sense to you. And at this point, let's take our break time with Gizmo and Wicket, see what these little guys are up to. We'll be right back to finish this chapter strong. Now, one thing I want you to know about microorganisms is that they live in different niches that they are adapted to. There are organisms that can live on my salty skin. You know, skin is a surface which has a number of microbes that live on it. And there are specific microbes that are adapted to living in this niche. These organisms can withstand my skin's conditions, right? And they feed off of oils in my skin. They can withstand the high salt, the low pH. They can withstand the environment. They can attach to my cells, you know. And so the same organisms that can live on my skin, not necessarily be able to live in my throat, right? Conversely, there are organisms that live in my throat that are adapted to living in the throat. So, for example, let me give you a good example. Did you know that if you get a cut on your skin, you're at high likelihood of getting a staph infection, right? So you could end up, if you don't clean out that wound, you might end up with a staph infection. You know why? Because staph prefer to grow on the skin, right? Whereas if I have irritation in my throat, or if I have nicks or abrasions in my throat, I'm susceptible to get strep infection in my throat, right? You know why? Because streptococci are more adapted to the niche in the throat, right? Whereas staphylococcus are adapted to the skin. Same thing with my intestines. E. coli would be inside of the intestines. Those are the niches in which they are adapted to grow. And those adaptations that allow you to live in these niches include temperature preferences. You know, some microbes prefer cool temperatures, some prefer hot. Oxygen requirements, pH requirements or tolerances, osmotic pressure tolerance, barometric pressure tolerance, right? So depending on which of these conditions a microbe prefers or can tolerate, that's where the microbe is likely to live. Isn't that neat? So when it comes to microbes, different microbes prefer different temperatures for growth. And when we look at those temperatures, there are 3 cardinal temperatures we look at for every different microbe: the minimum temperature, the maximum temperature, and then the optimum temperature. The optimum temperature is the temperature that promotes the fastest rate of growth and metabolism for the microorganism. They call this the preferred temperature of the microorganism. And no, it is not the same for all the different microorganisms. The maximum temperature for an organism is the highest temperature that it permits the microbe's growth and metabolism. Usually, if you go any higher than the maximum temperature that a microbe can tolerate, it will die, right? Because its proteins will denature and the cell will stop to grow. Here, you can see the minimum temperature. The minimum temperature is the lowest temperature that permits a microbe's growth and metabolism. Anything below the minimum temperature will not result in microbial death. It usually results in the microbe freezing, right? So at below minimum temperature, the microbe freezes. It doesn't necessarily die, right? Whereas past the maximum temperature, that's when the microbe stops working and falls apart. All right, so what do we call organisms that prefer to grow in cool conditions? Those are known as psychrophiles. See on this graph here, psychrophiles are in blue, and notice how they grow between negative 15 degrees and 15 degrees Celsius. Now, what types of microbes grow around room temperature or body temperature? Those are known as mesophiles. Mesophiles grow in, it looks like their mesophiles are the green line, and their optimum temperature, cardinal temperature, is around 37 degrees. You know what that means? Body temperature. I want you to write that down, please. 37 degrees C, you should know that 37 degrees C is body temperature, your body's temperature. And guess what? Certain mesophiles prefer your body's temperature, right? They prefer 37 degrees. Does that make sense? Whereas Psychrotrophs, see this orange line here? Psychrotrophs prefer, see their optimal is around room temperature. I want you to write down that 24 degrees is room temperature. Between 24 and 25 degrees C is room temperature, and that's the realm of the Psychrotrophs. And 37 degrees, that's the realm of the mesophiles. Does that make sense? So let me ask you this. Which of these is most likely to make a human sick, do you think? A psychrophile, a psychrotroph, or a mesophile? Which one of these three is the most likely to cause a human disease in your body? That's right, like it's always paying attention. Mesophiles, because they prefer body temperature for growth, they're the most likely candidates to cause human disease, right? Does that make sense? So for instance, do you think psychrophiles would cause human diseases? Look at psychrophiles. What's the maximum temperature that a psychrophile could grow at? 15 degrees? That's cooler than a room temperature, right? That's actually pretty cool. So guess what? Psychrophiles would not be causing human diseases. Now, you could get human diseases from a psychrotroph because, you know, right around body temperature is their maximum temperature. So possibly, but most likely what? It looks like mesophiles are the ones that are going to be responsible for human disease because those are the ones that like to grow at body temperature. Isn't that neat? And then it doesn't stop there. There are microbes that love to grow where it's really hot. I'm talking sulfur springs, hydrothermal vents, things like that. And here, these are called the thermophiles in pink and the extreme or hyperthermophiles in brown over here. I mean, that they can grow in conditions hotter than an autoclave. I mean, think about that for a second. So anyway, mesophiles grow best at body temperature. Psychrophiles grow best at very cool temperatures. Psychrotrophs grow in between the two, right? Isn't that neat? Next, let's talk about gas requirements, oxygen. Did you know that the earliest life forms did not use oxygen for growth? And it's an adaptation to even grow in the presence of oxygen. Isn't that neat to think about? Oxygen is vital for you and me to grow, right? And to live. But oxygen is actually quite toxic in living cells. When living cells use oxygen to grow, they end up converting oxygen to these weird oxygen variants that are highly dangerous to the cell. They're deleterious to the cell. So oxygen, when a cell is growing, the cell will convert oxygen to singlet oxygen, superoxide ion, peroxide, and even hydroxyl radicals. These are forms of oxygen. These are molecules of oxygen that when these are formed by living cells, they start destroying the cell, right? They're called free radicals, or ROS. ROS means reactive oxygen species, ROS, okay? And unless the cell has specific enzymes that are meant to deal with these radicals, then the cell ends up getting destroyed. Does that make sense? And so if a cell does not have the proper enzymes to break these back down into harmless forms of oxygen, then the cell ends up getting destroyed. And that's an organism that cannot grow in the presence of oxygen. However, if the organism does have the adaptation to produce those enzymes, like, look, superoxide dismutase and catalase, these are enzymes that are involved in converting these reactive oxygens back to non-reactive oxygens. And if that's the case, then the organism can grow in the presence of oxygen. Isn't that neat? So think about it this way. If the organism can produce these two enzymes, superoxide dismutase and catalase, it's likely it's going to grow in the presence of oxygen. If the organism can't produce these enzymes, so it doesn't have the genes to produce these enzymes, then it's going to get destroyed in the presence of oxygen. Think about it that way. And yes, there are some organisms that cannot grow in the presence of oxygen. There are some that only grow in the presence of oxygen. So let's talk about that. An aerobe is an organism that can utilize oxygen because it can detoxify oxygen. It probably forms the enzymes superoxide dismutase and catalase. So an aerobe can grow in the presence of oxygen. What is an obligate aerobe? It's a type of aerobe that must, when you see this word obligate, you should think must, right? Or vital. That an obligate aerobe must grow in the presence of oxygen. It cannot grow without oxygen. However, if an organism is a facultative anaerobe, that means it utilizes oxygen, but it can grow in the absence of oxygen. Did you know E. coli is an example of a facultative anaerobe? E. coli can grow outside of the body, but it can also grow in your intestines where there's very little to no oxygen as well. So that's an example of a facultative anaerobe. When you see this term facultative, think can or can't, right? Think with or without, right? So facultative anaerobes can grow with or without oxygen. They utilize oxygen. Obligate aerobes must grow in oxygen. Microaerophiles are kind of neat. These can grow in oxygen, but they only require a small amount of oxygen. So they tend to live in places with lower than atmospheric oxygen. Remember, in the atmosphere, there's 20% oxygen. These prefer to grow in slightly less than 20% oxygen. And what are anaerobes? Anaerobes are organisms that do not utilize oxygen. You know what that means? That means they probably don't have superoxide dismutase nor catalase. Does that make sense? So for instance, an obligate anaerobe, that means it must grow in the absence of oxygen. These guys lack superoxide dismutase and catalase, those enzymes to detoxify ROS, right, reactive oxygen species. So they cannot survive in oxygen environment. If you tried to grow one of these organisms in air, just regular air with oxygen, it would start building up those reactive oxygen species I told you about, and that would end up destroying the cell. Aerotolerant anaerobes are interesting. These are creatures that do not use oxygen, but they can still survive in its presence. So they probably do have at least one of those enzymes for mitigating the deleterious effects of ROS. Now, one way that scientists determine whether an organism requires oxygen or not is by inoculating. That means adding the microbe to this tube of solution called thioglycolate. This is a reduced solution, a reduced medium thioglycolate. That's the name of the solution inside of this tube. And this tube has high concentration of oxygen near the surface and about 0 oxygen at the bottom, right? So think about it as a gradient of oxygen from 100%, uh, air at the top, which means 20% oxygen at the surface and 0% oxygen at the bottom, right? So you inoculate these tubes with bacteria, and then you come back after you incubate to see where did the bacteria grow. So for instance, look at this. In tube 1, the bacteria only grew at the very, very, very surface. So what would you call that? Can you beat Wicket? What kind of oxygen requirement is that? That's right, Wicket. An obligate aerobe. Remember, obligate aerobe. These must grow in the presence of oxygen. So an obligate aerobe would only grow at the surface of a thioglycolate tube. Now let's look at this one on the far right, tube 4 on the far right. Notice how this one's not growing at the surface at all, okay? It's not growing where there's oxygen. There's only really oxygen in this part of the tube, really, if anything. So it's only growing at the bottom, but once the oxygen starts, appearing, there's no microbe, right? So in this case, this is an obligate anaerobe on the right. And the two in the middle are examples of what are called, remember, facultative anaerobes, right? Facultative anaerobes. Now, some microbes are strange. They require some CO2 in order to grow, and these are called capnophiles. Capnophiles grow best at higher CO2 levels. And did you know microbes also vary in the type of environment they prefer in terms of pH? So some microbes grow best near neutral pH. Remember, neutral pH is pH 7, but technically it's between pH 5.5 to 8. Neutrophiles are microbes that prefer neutral pH conditions for growth. Acidophiles grow best at extremely acidic pH. That means low pH, lower than 5.5. Alkalinophiles, by the way, another word for alkaline is base, right? So these enjoy basic environments. Alkalinophiles, these grow best in alkaline pH, right? So higher than 8. Now, some microbes can withstand osmotic pressure. I told you what osmotic pressure was earlier in the chapter, right? Remember, water either rushing into the cell or rushing out of the cell because of tonicity. Well, osmophiles, these are organisms that require a high solute concentration. And if it's salt that they require, it's called a halophile. Halophiles require high salt. So if it's an obligate halophile, that means it's an organism that requires high salt in order to grow. So a hypertonic environment of salt in order to grow. Whereas osmotolerant microbes, these don't require high concentrations of solute, but they can withstand high concentrations of solute. So for instance, if an organism is a facultative halophile, that means that it doesn't require high salt to grow, but it can withstand high salt. So an example is Staphylococcus aureus. Another environmental factor that can affect cell growth is pressure. Think about deep in the ocean where there's a lot of actual water pressure. If it's a barophile, it is able to grow under extreme water pressure. And by the way, water is required for all life. Remember, 70% of the cell's weight is water. So water is required for living things. Only dormant, dehydrated cell stages, such as spores, remember endospores, for instance, and cysts, these dormant stages of life are the ones that are, more resistant to dry conditions. But living cells cannot withstand the lack of water. All right, concept check number 5. Chlamydomonas nivalis is an organism that grows on Alaskan glaciers. That's what this picture is. It's an Alaskan glacier, and it's an organism That's photosynthetic pigments give the snow a red crust. That's why the snow looks red. This organism would be best described as which of the following? Well, wouldn't it be a psychrophile? Because that means grows in cold conditions. So yes, a psychrophile. A normal mesophile would not prefer those Alaskan conditions for growth, right? It would not be able to grow in the Alaskan environment. Now, organisms can also form nutritional dependencies on one another, or nutritional associations with one another. Discuss two of these microbial associations, symbiotic and non-symbiotic associations. So let's start with symbiotic associations. And this term symbiotic refers to organisms that live in close nutritional relationships that's required by either one or both of the members. So there are three forms of symbiotic relationships that you need to know about. Mutualism, this is where both members benefit. It may or may not be obligate, but both members benefit. So in this case, both of the organisms in the relationship, in the symbiotic relationship, benefit from the presence of one another. So I'll give you a quick example of mutualism where both members benefit. There's an example of obligate. Remember, this is a must. Obligate mutualism in termites. Termites rely on the endosymbiotic microbe, Trichonympha, for survival. See, you can see here those termites, and in the guts of the termite, you can see the Trichonympha. These are organisms that help the termite to digest wood. So the termite would not be able to digest wood if not for a variety of this endosymbiotic protist, right, the Trichonympha. And so it's a mutualistic symbiotic relationship, right? The termite eats the wood is broken down by the protozoa. The protozoa then releases those nutrients into the ant for the ant to grow, right? So they both require each other for nutrition. This is an obligate symbiosis. An example of a non-obligate mutualism, they benefit from each other's presence, but it's not required, right? So an example is the Silophorin euploites, which is a unicellular green algae. See, this Silophorin, it's a ciliated protozoa, and it harbors these green algae inside. Isn't that neat? So it ingested the green algae, but it does not destroy the green algae. Instead, the green algae now have a nice place to live. The green algae can produce sugar through photosynthesis, which the ciliate can feed off of. And the ciliate provides a nice environment in which the algae can grow. Again, they don't require each other. They do have mutualistic benefits when combined. Now, another type of symbiotic relationship is called commensalism. In commensalism, the commensal organism benefits. The other member neither benefits nor is harmed. Here's an example of commensalism. Again, during commensalism, the commensal benefits. The other member is neither harmed nor benefits. Staphylococcus that grows on your skin, right? Think about the staph that grows on your skin. So take a look here. This is Staphylococcus that grows on your skin. It's not actually benefiting you. The staph that grows on your skin doesn't do anything for you, but you're helping it out because it can feed off of those oils inside of your hair follicles and on your skin. So the staph's benefiting, but you're neither benefited nor harmed. The same thing with the mites that live on your eyelashes. Did you know that you have mites in and around your hair follicles called demodex? Demodex are mites that live in your hair follicles. And this is another example of a commensal organism. And this mite that lives in your hair follicles, it's benefiting from living there, but it's neither harming nor benefiting you. And by the way, I think I mentioned this was Staphylococcus, but here it says it's Micrococcus luteus. That's another example of an organism that lives on the skin. So staph, or either Staphylococcus or Micrococcus, either way, if an organism like this lives on your skin, it's not really benefiting you, nor is it harming you. So that's an example of a commensal relationship. Now, this last type of symbiotic relationship really trips students up because they don't think of it as a symbiotic relationship. When people think of symbiotic relationships, they think of, everyone's helping each other out. Remember, symbiotic simply means organisms living in close nutritional relationships. It could be friendly or it could actually be adversarial. So for instance, parasitism is a type. Parasitism is a type of symbiotic relationship where the parasite is the organism that becomes nutritionally dependent and benefits upon the host cell or the host itself, and the host is harmed. Right. So again, with parasitism, the parasite becomes dependent on the host and benefits, while the host itself is harmed. All viruses are parasites. So for instance, look at this. This is an image of the hantavirus, which is a human pathogen, human parasite, where the virus benefits while the human is harmed. Malaria is a disease caused by a parasite, right, called Plasmodium, which is a type of protozoa. Here you can see that through mosquito bites, the Plasmodium parasite is spread from person to person, and this parasite is becoming nutritionally dependent on the host, right? So the parasite benefits, but the host gets harmed and forms malaria. So that's it for symbiotic associations. Now let's talk about a form of non-symbiotic association. In this case, with non-symbiotic, these are organisms that are free-living, that have relationships not required for survival. So in this case, here's the example of non-symbiotic relationship I want to tell you about. It's called syntrophy. This is interesting. This is where members cooperate and share nutrients. Syntrophy is also known as cross-feeding. Here's where this is interesting. This is where microbes sharing a habitat feed off of substances released by other organisms. There's a nice example here with Azotobacter and Cellulomonas. Here you can see the association between the two. Azotobacter is an organism that's a nitrogen fixer. Remember what that means? Takes nitrogen out of the air and fixes it into actual molecules, right? Like, for instance, ammonia, or into proteins. And then this organism takes nitrogen out of the air and it produces NH4+, which is ammonia. Then the Cellulomonas takes the ammonia from the environment and it produces glucose. And then guess what? The Azotobacter uses the glucose. So the product of one organism is used as a nutrient in the next organism, and then the product of that organism is used as a nutrient in the first organism. You see how this is a relationship based on nutrient cross-feeding? That's what that means. Another form of microbial relationship occurs in what are known as biofilms. Biofilms are really neat communities that microbes make. And remember, in biology, a community includes members of different species, right? Many different species make up a community. So biofilms result when organisms attach to a substrate, that means some sort of surface, by some form of extracellular matrix. Think of glycocalyx. Remember the sugary proteinaceous goo that bacteria can secrete? That binds them together in complex organized layers. This is the dominant structure of most natural environments on Earth. In fact, if you've ever been familiar with plaque on your teeth, right? When you don't brush your teeth, it kind of feels gritty. You form that plaque. That plaque on your teeth is an example of a biofilm, right? Many different organisms growing together in that goo, right? Think of it that way. And what's neat is that not only do these microbes grow in that community, but they can communicate and even cooperate in that community by a process known as quorum sensing. So this is how biofilms form and how quorum sensing works. First of all, step 1, free swimming cells settle down and attach to the surface of some substrate. Let's pretend this substrate down here is your tooth enamel, right? So the microbe might attach to your tooth enamel with its little fimbriae. Remember, its bristles. So you've got these organisms that now attach to a surface. Step 2. Once attached, these microbes can secrete some glycocalyx, some sugary proteinaceous goo that helps to keep them on the surface of that substrate. Step 3. Each one of these microbes releases a molecule, this depicted here as a blue circle. These blue dots represent what are known as quorum molecules. And these quorum molecules build up in concentration. The more bacteria are in this biofilm, the more quorum molecule builds up. Step 4. Once these quorum molecules are concentrated enough, they can enter a bacterial cell in the biofilm and turn on various genes. In this case, the quorum-dependent gene produced this quorum-dependent protein. So this protein was made only because there was a high concentration of these blue dots in the environment, these quorum molecules in the environment. That means that there's sufficient number of bacteria to produce those quorum molecules. Does that make sense? So if there aren't many bacteria in this biofilm, then you're not going to produce enough of this blue quorum molecule to turn on these genes to produce these proteins. But in this case, the protein was made, and then those proteins can work to help digest food particles, for instance. So in this case, the quorum protein is helping to digest food for the rest of the community here. Isn't that neat? So think of a biofilm as a gooey substance in which multiple different microbes grow and they kind of synergize and they work together. And think of quorum sensing as a way for microbes to communicate and coordinate. That's because they can release that molecule, the quorum molecule, but that molecule doesn't have an effect unless there's a certain number of microbes in the community. Now let's talk about interrelationships between microbes and humans. The human body is a rich habitat of symbiotic bacteria, fungi, and a few protozoa. This is known as your normal microbial flora or microbiota. In fact, did you know that you have more microbes that live on your body than you do your own number of cells? So that's nucleated cells anyway. So isn't that neat that you actually tote around on your body more microbes than you actually have your own nucleated cells? Think about that for a second. Some of these organisms are commensal, some are parasitic, some are synergistic. They can affect our physiology, our immunity, and even our genetics. So when we talk about microbial growth, they've got the nutrition, so they're growing. Microbial growth occurs at two levels. Growth at the cellular level with an increase in size, so the cell could grow in size, but also the cell could divide to increase the population. So for instance, 1 E. coli could divide to give you 2 E. coli. And do you remember that division of bacterial cells occurs mainly through a process known as binary fission? And remember, that this is a form of asexual reproduction, where a single cell, like E. coli, divides by binary fission into 2 E. coli. During this process, the parent cell enlarges, duplicates its chromosome, remember its nucleoid, and forms a central transverse septum. The septum is the way that bacteria undergoes cytokinesis. It's through septum formation, and then divide into 2 daughter cells, 2 genetically identical daughter cells. So again, these are the steps to binary fission. Let's look at step 1 at the top. A parent cell at the beginning of its cell cycle. What cannot be seen is the synthesis and activity gearing up for cell division. So this cell grows in size. Next, the cell decides to divide. So the chromosome is replicated. Remember, this is the circularized chromosome. I told you guys that bacteria have a single circularized chromosome called the nucleoid. Well, that nucleoid copies itself, and now you have 2 copies of that nucleoid. You have 2 copies of that circularized DNA. These are identical copies of DNA. And so, again, during step 2, you copy the chromosome, and then those chromosomes move to opposite ends of the cell. And notice what happens at this point. As those 2 chromosomes move to opposite ends of the cell, a septum forms. Remember, at the center of the cell, the cell wall begins to synthesize or build inwards. The cell wall builds inwards, and that's called septum formation. And that's the process by which bacteria undergo cytokinesis, the separation of the cytoplasm and of the cell. Step 4, completion of cell compartments. The septum is synthesized. You see, now the septum has fully formed, so you have 2 distinct daughter cells. And each daughter cell has a membrane around the cell. And then, of course, step 5, end of cell division. The daughter cells are now independent units. You have 2 genetically identical daughter cells. So now let's talk about how fast microbes divide. That's called the rate of population growth. The time that's required to complete one binary fission cycle is called the generation or doubling time. Each new binary fission cycle increases the population by a factor of 2. That means each time the cells divide, you have twice as many cells, right? So that means that you have exponential growth with the base of 2. Generation times for microbes can vary from a few minutes, like for instance, E. coli can double roughly every 20 minutes, to a few days, right? Some organisms take days to double. Here you can see the rate of population growth starting with a single cell. After 1 generation, you have 2 cells. After 2 generations, you have 4 cells. After 4 generations, you have 8 cells. Notice that each time you have a generation, you double the number of cells from 1 to 2 to 4 to 8 to 16 to 32. In the second row, you can see the number of generations. And in the third row, you can see how you can calculate the number of cells. If you start with 1 cell, then you have 2 to the power of 0 cells. That's basically, you always have a base 2. At time 0, you have 0 generations, which means you have 2 to the power of 0 cells, which means you have 1 cell. After 1 generation, you have 2 to the power of 1 cells, which means you have 2 cells. After 2 generations, you have 2 to the power of 2 cells. 2 to the power of 2 is 4. After 3 generations, you have 2 to the power of 3 cells, which means you have 8 cells total. You see, so the equation for calculating total number of cells is the exponential value 2 to the n. Now, what's neat is on a logarithmic scale, this is linear, right? So You see the dashed line shows you logarithmic growth over time. But if we're looking at raw numbers of cells, that's the dark pink line. The dark red line is showing you how many cells you have over time. The dashed line shows you the log of that. So when bacteria grow, they grow linearly in a logarithmic fashion. However, if we look at the raw numbers, we have a curve that goes asymptotic. So do you know what that means? That means that bacteria might seem like they start out pretty slow, but once they get going after a few hours, the numbers explode, right? And so you can depict this either by a logarithmic line or by looking at how the bacterial numbers go asymptotic over time. So this is why bacteria are interesting, because of exponential growth. In the lab, sometimes we will plate 1 or 2 cells. We'll come back in just 3 days, and there may be millions or billions of cells where there were originally 1. And that's because, like I said, if the bacteria are doubling every 20 minutes, then it doesn't take but 5 hours to have a million bacterium. Isn't that neat? So think about it this way. It takes 5 hours for one bacterium to become a million. Now, let me see if you could beat Wicket. Let me try to put your thinking cap on here. Let's see if you can beat Wicket. If it takes 5 hours for one bacterium to become a million, how much longer would it take to get your next million if the doubling time is 20 minutes? Can you beat Wicket? That's right, Wicket. He's so smart. Just another 20 minutes. Isn't that mind-blowing? It took 5 hours for one bacteria to become a million. But guess what? They're doubling every 20 minutes. So after only another 20 minutes, at 5 hours and 20 minutes, you'll have 2 million. And then 20 minutes after that, you'll have 4 million. And then 20 minutes after that, you'll have 8 million. So before you know it, you'll come back to lab, and your single little bacteria that you plated on a little agar plate is now billions of bacteria. And that's what formed little colonies on your plates. Isn't that neat? So the exponential growth is not to be, underestimated. Exponential growth is so powerful. It may start out slow and not impressive because one bacteria is becoming 2, 2 bacteria becomes 4, 4 bacteria become 8. That's not very impressive. But once 1 million becomes 2 million, 2 million becomes 4 million, and then it goes asymptotic, that's when we're talking about billions and trillions of cells in no time at all. And again, this is the equation for the rate of population growth, where NF is the total number of cells in the population, Ni is the starting number of cells, and then you multiply that by 2 to the power of n, where n denotes the generation time. This is the equation for determining total number of cells in a population. And with that, it leads us to concept check number 7. Escherichia coli, E. coli, has a doubling time of 20 minutes. If there are 5 cells at the beginning of the experiment, so we'd have to plug in here, right? Ni is 5. Ni is 5. How many will there be in 3 hours? Remember, 3 hours means 3... and there's a doubling time of 20 minutes, right? There's a doubling time of 20 minutes. You started with 5 cells, and so you need to figure out the generation time. Does that make sense? The generation time is what? The generation time was 20 minutes, right? 20 minutes. But you left it there for 3 hours. So how many blocks of 20 minutes? Let me put it this way. How many 20-minute stretches are in 3 hours? Can you beat Wicket? How many 20-minute stretches are in 3 hours? That's right. Wicket is always 9, right? 9 20-minute blocks within 3 hours. So 5 times 2 to the power of 9 is your answer. So can we solve that? What's 2 to the power of 9? 2 to the power of 9 is 512. Times 5 is 512 times 5 equals 2,560. That's D. Okay, I hope that made sense, but remember, you need to use this equation in order to determine the answer to that kind of question. And then the last part of this chapter, we're talking about population growth inside of what's called a batch culture. You know what a batch culture is? It's a type of bacterial environment with limited space and resources, right? So imagine like a flask. If I have a flask of broth, isn't that limited space and limited resources in that flask? Well, that's what they mean by batch culture. So when we're talking about the growth curve, we're talking about the growth curve inside of a batch culture, inside of a limited space, limited nutrient environment. So when you put bacteria in such an environment, there are certain phases that occur. The lag phase, the exponential or logarithmic growth phase, the stationary phase, and then the senescence or death phase. So when you add bacteria to a broth, the lag phase is a period of little or no bacterial growth that occurs. And the reason for that is because the bacteria are adjusting to their new environment. So you should say during the lag phase of batch growth or growth curve, very little bacterial division is going on. Then the bacteria enter the exponential growth phase or the logarithmic growth phase, where they are truly growing logarithmically. Remember, 2 become 4, become 8, becomes 16, become 32, and so on and so forth. Then at some point, the party's over, right? So that means that they've run out of space or resources or nutrients. And so the bacteria enter a stationary phase where they are no longer doubling. And I should have mentioned this earlier, but on the y-axis, you have number of living cells, number of viable cells. On the x-axis, you have hours. So in the first 5 hours, you have the lag phase. Not much bacterial cell division going on during the lag phase because it's adjusting to its new environment. Next, you enter the exponential growth phase, and that means that you're truly doubling every generation time. And then you enter the stationary phase when you've run out of some kind of important nutrient. And then over time, there is the death phase or senescence phase. And that's because bacteria, they've run out of nutrients. They're kind of floating around in their own debris and their own waste. And so they start to die off. Isn't that neat? So that's called the stages in a population growth curve in batch culture. And also in the lab, we can tell how much bacteria is growing inside of a broth through turbidometry. We can look at 2 broths. So look, here's fresh broth on the left, and here's bacterial culture in the broth on the right. Notice how the culture becomes cloudy, right? You see, fresh broth with no bacteria is clear, not colorless, but clear. Here you can see that the broth has become very cloudy. That's called turbid. We can place these tubes inside of a spectrophotometer, which shines light through the specimen. The more light makes it across the specimen, we know the less cloudy it is, which means the fewer bacterium there are. The more light is blocked, the more light is A for absorbed, right? You see, the more light is blocked, your absorbance is higher. That means there are more microbes in the solution. The less light is blocked, that means your absorbance is lower, which means there are fewer microbes in the solution. So by measuring turbidity with turbidometry using a spectrophotometer, we can roughly figure out how many bacteria are inside of a broth culture. And that finally leads us to the end of this chapter. A very interesting chapter. I hope you learned a lot. Please leave any comments or questions in the comment box below, and I will catch you guys next time.
Dr. D, Dr. D, Dr. D. 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 A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D.